Thank you for joining me this afternoon to have a conversation about biocompatibility. Today, my discussion is really going to focus on what's changed in the standards. This realm of medical device has really become a hot topic lately, and hopefully you'll see why with the changes I'll present today. So if we look at this slide, you can see over the last 10 years, we've had 9 standards update. And a couple of these updates have been incredibly significant, which changed the way we approached biocompatibility and actually changed the way we were regulated for that as well, which drives the behavior here. So you can see we have 10993 Part 10, Part 12, 3 and 5, 4, 11, 6, 1, and 18 that have all updated between 2010 and 2018. Pretty significant, or 2019, pretty significant changes here as well. So I'm just going to dive right in and go through each one of these standard changes and talk about what impact that could have had to the testing that was performed maybe under previous standards, and also what I'm seeing that's different in the regulatory realm of things as well, that may not be different in the standards, but I'm hearing different feedback from regulatory agencies that I want to pass on to you as well. So if we look at 10993 Part 10, which is irritation and sensitization, the biggest change that happened was an increase actually in the number of animals that were to be used. This is kind of backwards from where we're trying to go, right, in the industry, where we're trying to reduce animal testing. But in this case, it's important from a statistical analysis standpoint. So we like odd numbers when we analyze data because it breaks any ties that we could see. So asking for a third animal is actually appropriate in this situation. So how does that impact your testing? If you had testing done for irritation prior to 2010, you want to look at the actual results from the animals to see if there was a discrepancy in what they were exhibiting as far as an irritation response. And then write a justification either that no difference was seen, so a third animal, would not provide useful data, or there was a discrepancy seen here. We need to verify this result. That doesn't necessarily mean you have to step into testing, but you do want to think about whether that result can be considered valid under the current standard requirements. So this is just an outline of what irritation testing looks like now. So we do use 3 rabbits. There's 10 injections for each rabbit, 5 of the control article and 5 of the test article. On each side, we do run this test in 2 extracts, so a polar and a non-polar solvent. So that's why we do request that we have 2 samples as a minimum, so we can have 2 separate extracts. It does take 4 to 5 weeks. It's about, you think about 3 days for an extraction, and then we observe the animals over 3 days. So it's about 6 days of actual testing, but we have to get the animals ordered, have them acclimated, run the test, and then report the results. So that's why the test goes for 4 to 5 weeks. So there are further changes coming to this standard actually right now. I just went to the ISO meetings last week for this standard, and we are changing this now, but until that comes into effect, we're still following this guidance where we're talking about still doing in vivo testing. So if you have questions about the upcoming changes, which will be published next year, please feel free to talk to me afterwards. I'd love to talk to you about those changes. Okay, in 2012, we had an update to 10993 Part 12. This is an interesting standard because it affects all of the biocompatibility tests, because this tells a laboratory how you have to prepare the samples and run the extracts. So any changes to this standard can be very significant for the test process. So you can see here, just from a quick summary of what actually changed, some additional definitions for types of extractions that we can run. You'll often hear in the standards or in conversations with regulatory agencies that they're asking for exaggerated or exhaustive extractions. And if you want more information about what that actually means from a testing standpoint and what you need to have as a conversation with your laboratory, you can go to 10993 Part 12 and look at those definitions. You'll hear most of the time we want to do exaggerated extractions. But for like chemistry and genotoxicity testing, where we need some really sensitive numbers for that analysis, we're going to go more towards exhaustive extraction. So just things for you to keep in mind to do some more research about your products and your testing, and keep those terms in mind. I want to talk a little bit about what I actually see that's a discrepancy in testing that causes companies to actually have to retest their biocompatibility, and it's what actually was tested. So not only are the standards updating over time, but the device is changing over time. So when you go back and you say, okay, let's look and see if our biocomp is current, you also have to look at what device was tested and what device is actually being marketed, and are they the same? And when I say the same, I mean exactly the same. Same supplier, same material, same colorants, same ratios, same geometry. If those things have changed over time, then that needs to be included in the gap analysis that you'll need to run to determine whether that testing previously done is still appropriate. What I also see, especially, which is relevant for a trade show like this, is where we do a lot of analysis on raw materials, and is that going to translate to a finished device? We have to understand and evaluate the processing effects. That cannot always be done by a literature search. Sometimes you do actually have to gather data and step into testing. So you have to think about any additives, any colorants, what kind of sterilization is used. So if you use testing from a raw material manufacturer that tested the material non-sterile, but you're going to use gamma sterilization and add a colorant, those are not the same materials. So the testing cannot be directly correlated to protect patient safety. So that's definitely something that I see a lot of times. And I want to point out what's given to us in guidance in 10993 Part 1 specifically, that says you must evaluate a final finished device or a representative sample of this final finished product. It can be a coupon, it can be what we call a master device, but you have to line up where it's incorporating not just materials, but also processes. Another thing in testing that I see as a discrepancy is how was it actually tested? Did we use surface area or did we use weight in the laboratory setting? And you may think, well, that's the lab's job to understand that. But actually, as the manufacturer and the requester of the testing, you are in control of what's actually happening. You're the central command center. So you have to understand how the device is being tested so that it remains consistent throughout all communication with testing to your laboratory. So if you tested by surface or by weight originally, I'll tell you that's not preferred any longer. Even though it's still allowed in 10993 Part 12, we're seeing submissions be questioned when the testing is done by weight. And I'll give you an example of why that's the case. So this is a permanent implant. You can see that we have metals and polymers here, so fairly dense material for most of the device. So if we're going to test this by weight as a laboratory and using the standard guidance, we are to use a 0.2 gram per milliliter ratio according to 10993 Part 12. So when we think about the weight of the device, we apply that ratio, we end up adding almost 500 milliliters to this sample for the extraction. Still allowed in the standards and could be considered appropriate if we just followed letter of the law. If we look at surface area, this device is actually quite small. It's about this big. So surface area wise, it's not even quite 120 centimeters squared. We apply another standard ratio according to 10993 Part 12, which is the 3 centimeters squared based on sample thickness. And that leads us to only adding 38.6 milliliters. So when you look at that and you want to think, what's the most appropriate extraction from a scientific standpoint, we want to concentrate that extract as much as possible. So we want as little fluid per device as possible because we want to pull everything off we can and then test as much of that solution. So comparing weight versus surface area, in this case, that ends up being a 12-fold dilution. And I'll tell you, Clorox will pass any test at a 4-fold dilution. So we're not going to detect anything off this metal device if we do it by weight. So just another thing to keep in mind, while the weight ratio is still allowed in the standards, it's not preferred by any regulatory agency. They're going to question a submission based on weight for testing. Another thing that needs to be considered is at what temperature was it extracted at? This is one I've actually seen last week from the FDA for a company where they did a 37-degree extract. So right now, these are all still parameters allowed in 10993 Part 12, but in the industry, it's standard practice to only use 37 degrees for cytotoxicity testing. We have to do that based on the media that we use for the extract. Any higher temperature will actually denature the proteins in the extract, so we're not able to use that. So we keep at 37 degrees. Typically, it's a 24-hour extract, but the FDA has come out and said, if you have a permanent implant, we now want to see 37 degrees for 72 hours. And that's a brand new one by the FDA. So if you are submitting a permanent contacting device and your cytotoxicity was performed at 24-hour, be ready for the question or go ahead and repeat the testing at the preferred extraction time. The exaggerated extraction that's preferred right now is a 50 degree. So again, 37 is appropriate from a patient protective basis because it's basal body temperature, but we want to exaggerate, again, the exposure conditions. So that's why we prefer a 50 degree for 72 hours. So just things to keep in mind when you're reviewing your previous data. Okay, in 2014, the cytotoxicity standard was updated. This is 10993 Part 5. Now, in this revision in 2014 and the one previous, we already had identified a pass-fail criteria. This comes from the USP biocompatibility standards. So a score of 2 or lower is considered passing. If you are higher than 2, scoring a 3 or a 4, that is considered failing. What's changed between the previous standard and this 2014 is a more preferred preference for a quantitative assay, something like MTT or XTT analysis. So MEM elution, which is the standard cytotoxicity test that has been used for decades, is what we call a subjective test. It's qualitative. So it's based on an analyst looking into a microscope and making a judgment call according to this scale, seeing and deciding if they see 50% cell death, if they see 10%, if they see 100% cell health. It's all based on their interpretation of what they're viewing and how they can gauge that percent population health. So there is some discrepancy here in that. However, you do gain the benefit of having an actual analyst observe this to look at overall cell health and other observations that they may note here. So in the standard, with the preference put on a quantitative assay for final finished medical devices, I like to highlight the discrepancy in a passing criteria. If any of you have performed cyto, I guarantee more than half of you have seen a failure in that test. It's an incredibly sensitive test. It is also not compatible with a lot of things we put in medical devices like antimicrobial agents, latex, rubber, inks, things that are okay really in a clinical setting, but will not work in the in vitro analysis here. So it's already a very sensitive test. If we move to this quantitative assay, we've also then increased the sensitivity of a test that's already very sensitive and doesn't necessarily represent the patient exposure or protect patient safety in the clinical setting. So I would just ask, if you're looking at the standard and you're reading it for the letter of the law and you say, oh, we have to move to this MTT, if you're already sitting at a 1 or a 2 in your cytotoxicity test routinely, moving to MTT is probably going to push you up to a failure. And will it protect patient safety? So there's just some thought and discussion about the appropriate method that should be used that needs to be considered here with some additional justifications. Okay, also in 2014, we had an update to the genotoxicity standard. So there's not a lot here that actually would affect the what's done in testing. More exhaustive extraction details were actually added to some of the annexes, and this is based mainly on from Japanese requirements. So my Japanese counterparts that work on the ISO committee are very concerned about our extracts that we use for genotoxicity testing and like to see those extracts as concentrated as possible. So those are considerations. We're actually revising it again right now. So a new 10993 Part 3 will come out in the next year and a half, which also clarifies these extraction expectations. But I'll tell you what I see most often in my data review is a consideration about what genotoxicity tests should be done. The 10993 Part 3 standard does ask for 2 tests to be performed. So even though it's one criteria or one biological endpoint that's mentioned in 10993 Part 1 here, where it will say genotoxicity is necessary, it actually is a 2-part requirement that has to be filled. So you need to do An Ames test is the preferred bacterial test that should be performed. And then they're asking for a mammalian cell test to be performed. So I've listed some options here. There's several other options, but these are more common in the industry, with chromosome aberration and mouse lymphoma being the most common performed mammalian tests in the industry. So in my review, what I often find is that only Ames testing was performed. And when submissions are sent again, they're being told, oh, your genotoxicity requirement has not been sufficient because you've only performed one test. So again, not a change in the standard, but it's something I've noticed in my review of previous testing that this category's been incomplete. Okay, in 2016, there was an update to the implantation standard. And again, not a lot here was added that would address or apply to a lot of device manufacturers, some more specific local implant criteria given especially for like a brain tissue implant. However, one thing I see again in when I review past reports is what was used as a negative control. This is a very important consideration for this test, especially if you have a product that is meant to have tissue ingrowth. So if you have a product that provides scaffolding later for tissue to ingrow, because that works better for the patient, you need to have a comparable control for that. So you need to keep the standard negative control for this test, which is HDPE, which is going to show no tissue ingrowth whatsoever. And then you also have to show a control of something that's already currently approved and on the market as a control, and then your device as the test subject. And this has been specifically laid out by the US FDA because they want to see that your tissue ingrowth response is similar to something that's already out on the market. This is because when you look at the results, any tissue ingrowth would be categorized as an irritant. So you would fail the test automatically compared to a negative control. So that's why you want this comparison device to say, hey, we have tissue ingrowth, but it's appropriate, it's intended, and we can have data to show we're very similar to something that's already in use. Okay, so just additional, again, things that haven't changed in the standard, but I see being raised as questions by regulatory agencies. Okay, in 2017, 10993 Part 4 updated. And I would say this is probably one of the harder standards to get complete agreement on in the regulatory realm of things of how they particularly expect it to be addressed. Even though the FDA says on their consensus standard website that they approve 10993 Part 4 in whole, we know from conversations that their expectations really of how devices are assessed is a little bit different. So this is just Biocomp according to Audrey 101 here, where I've identified based on our experience in conversations with FDA, we typically see this very general approach to hemocompatibility from the FDA. I can't speak from a notified body perspective just because of everything that's happening with the MDR and that more than half the notified bodies went away. So this is specifically about the US FDA right now. So for direct contact with circulating blood, hemolysis complement thrombosis analysis, indirect contact, only indirect hemolysis, and then devices with shear stress, you have to have a dynamic hemolysis. We call that a mechanically induced hemolysis test. And then one thing that the FDA put in their guidance document, when you assess complement, they only expect to see SC5 beta 9. We don't have that specified in the ISO standard, and we haven't heard from notified bodies if they're still requiring a second protein analysis. So again, additional things to keep in mind as you're reviewing your testing. And if you have hemocompatibility data that needs to be reviewed, I would highly encourage bringing in some experts to review that and give you some advice because it's a very complex set of tests to address. Okay, also in 2017, we had 10993 Part 11 update, which deals with systemic toxicity tests. So this covers a wide range of tests actually that are listed in the table in ISO 10993 Part 1, where it covers acute systemic toxicity, subacute, subchronic, chronic toxicity, and it also addresses material-mediated pyrogen testing, all systemic response-based tests that we're looking at. So the major difference here is that they actually lowered the number of animals that they're suggesting for chronic toxicity testing. So no, not necessarily a repeat in testing, But if you did a lower number originally and didn't meet the 40 previously, then you should be okay here for this endpoint. Okay, in 2018, one of the more significant standards was changed, and that is 10993 Part 1. So this is what we call, this is like the umbrella standard. So this talks about all of biocompatibility and how we're adjusting it for every medical device. And there were some pretty significant changes here. And just as a summary, there was a revision to Table A1. This is like the go-to chart for biocompatibility that we all look at. We're going to go over those details and specifics, but they've actually changed the conversation. They changed the way it's written so that we have different conversations about the testing and assessments that are required for medical devices. And that's with this inclusion of X and E, and we'll talk about that specifically. Also, they've replaced Annex B, and they've included some fabulous guidance on how to do a risk analysis. So that's really important because that's where all of your documentation needs to come from, is starting from this risk-based analysis, and they give great guidance in this annex. And then there's new definitions, and that will be very helpful for your analysis as well. So let's talk about the table and what's changed. So if we look here, the first thing is this requirement for physical and or chemical information. You'll notice there's an X in this column for every single type of device. Now, the one thing I like to say right off the bat is this does not mean you have to run and do full extractable and leachables testing. That is not the implication here. What they're looking for is that you actually identify what materials, what suppliers, what processing is used on the device so that you can understand how your device is made before you put it into testing. So as that device changes, you can track that a little bit better. Okay. For whatever reason, we cannot get every blank completed in this table, no matter how hard we try. We left a blank last time. We thought we got them all covered. This third column should be sensitization. So that's an easy assumption. It's the one missing. It should be sensitization. Material-mediated pyrogen. This is not a new test requirement, but they've put it on the table to highlight that it is a requirement to be addressed. This is different from bacterial endotoxins, which are also a pyrogen source, but it's from endotoxins. We're talking about pyrogens that come from different sources, from the actual materials you add into the device. ISO 10993 Part 11 has an annex which includes a list of known material pyrogens, so you can review that list to give you some more insight to what's expected here. Okay, this column for subacute and subchronic, those are not new tests. They were included in the previous table before, but they're actually split now. And you'll see as you look down the table that for some device contact time, they're only looking for subacute, not necessarily a subchronic testing endpoint, which is important because it saves you time as you're doing your biocompatibility. These are very, very long tests, so it saves you a lot of time. Chronic toxicity is also new to the table, but not new to the conversation. But again, it's being put in the table to highlight that you need to address this and think about this endpoint for your device. And then these last three, carcinogenicity, degradation, and reproductive and developmental toxicity. You'll notice there's not an E for any medical device here, because this could apply to so many medical devices not based on contact duration or type, right? And depending on your patient population and the intended function of the device, if it's meant to degrade over time and become absorbed in the body. So you have to think about what is a function of our device? What's our patient population? So thinking more outside the box. Coming to this table as a checkbox approach does not work. You still have to start with your risk-based analysis, which starts with the device contact, time, and type, and then moves into your patient population and then goes down from there. One thing I want to highlight is you can see here that this change in this table, these changes, just identifying the new columns, is not all that's changed, but they've actually added more test requirements for these two test categories, or these two device categories in particular, mucosal membrane and breached or compromised surface area. If you are a wound healing device manufacturer, you're probably feeling a little bit of biocomp pain right now because the regulatory agencies have put the heat on this market in a huge way, asking for additional assessment of the products. So there's some significant changes here and creative approaches to the actual testing that needs to be done, because a lot of these products contain antimicrobial agents which won't pass cytotoxicity. They also are bioabsorbable. So they lend to be very complicated. And you would think the higher risk devices are more complicated. That is not true. Higher risk devices have a very straight path of what's expected. It's when it's a more difficult risk to actually assess that we have a little bit more trouble, clarifying that path and working that out with the regulatory agencies. This is the second half of that table. It does go on to 2 pages, and I brought this up to highlight specifically the X and E. So you'll see X is required for every single medical device. So physical and or chemical information does need to be gathered. And the E does not mean you have to do testing, which is great. And you wouldn't expect somebody from a laboratory to say that, but I think it's great if we can save some animal lives and use data that we already have. So this allows you to step into a conversation with the regulatory agencies and say, hey, look, we have this other product that's identical except for maybe geometry, and it uses all the same products, all the same manufacturing, and we've fully assessed that for biocompatibility. You get to really use that data now to support your biocompatibility from a risk-based perspective. So I love that the E is there because it does allow a different conversation to be had where you can use what you already own as a company. So again, one of the changes to -1 was this inclusion of Annex B, which goes into a risk analysis, risk assessment discussion. I really recommend, if you've been familiar with the table maybe part of 1093 Part 1, to look at this because it does give you a clue about what the regulatory agencies, notified bodies from around the world are expecting as far as the conversation. And I would say specifically when you are using previous testing information from previous devices, just saying it's the same does not cut it. You really have to show out by your processes with a comparison table to really show that everything is exactly the same and highlight the differences and then address those differences. So the definitions that are added, I'm just going to spend a little bit of time talking about this because maybe you've heard in the industry discussion about chemical characterization, extractables and leachables, a material characterization, physical and or chemical information. These terms are all used often interchangeably, but they mean very different things from a risk assessment perspective. And it's important that you're careful when you write your documents that you clarify those points to show that you are adhering to the standard and that your testing has been set out in an appropriate way. So I'll give you some examples, but just a minute to review the material characterization, physical or chemical information, and then we'll talk about this transitory contact. So the first 2 definitions really help us address that first category of requirement where it says physical and or chemical information. Now, I would say there's some misleading parts to 1093 Part 1 that were not caught in the last review that tell you you have to consider testing according to -18, which is full-on chemical characterization. We're talking very extensive chemistry that takes weeks, is very expensive, and then has to be analyzed by a toxicologist and through a toxicological risk assessment. That is not needed for every single medical device because it will not always protect patient safety. We can't always get the right information from that data. So we want to start with the end in mind. Think about what data are you going to get from the testing and how is that going to apply in the end to the patient, which is what we're trying to assess here. So we want to keep that in mind. So we start with gathering the physical and our chemical information. And then we think about what kind of patient contact our device has. If this is a surface contacting device that is used every day, all day long, taken on and off, I would tell you the chemical characterization testing will not assess patient safety because we're not talking about something that's ingested, metabolized, and that becomes part of the biological system. We're talking about irritants, sensitizers. Those are the things we're going to have a concern about from a surface contact standpoint. So those are the conversations you have to step into when you say, this is how we're going to approach our material characterization, and this is how much more testing we need from a chemistry standpoint to understand our materials, or we understand our materials, and now we need to talk about patient safety. So use these definitions, read 10993 Part 1, because it really does give some good guidance on how to have that conversation. I love this third definition of transitory contact because it allows us to, again, assess true patient risk. There's a discrepancy, though, between the US and the EU on how they're considering transitory contact. So FDA put this definition, similar to this, in their 2016 guidance on how to use 10993 Part 1. They gave an example of a device, a hypodermic needle, used for less than 1 minute. Seems very logical, low risk to the patient. The needle material is a stainless steel used in the industry for decades. We're not expecting severe patient response. We do not need to do a huge series of animal tests to protect a patient here. But the EU gave an example of less than 1 hour patient contact. So if you're submitting to both markets, be careful in your definition here of what type of contact you're going to consider your device. You'll notice, though, transitory contact is not listed on the 10993 Part 1 table. We'll go back to that. It's not listed as an option of contact because right then and there, when you talk about patient contact, if you've identified that it's transitory, you're already going to have a discussion. Do we need to step into testing just based on this risk? Where is it touching the patient? Okay. So, and if you're in high risk areas like potential contact with cerebral spinal fluid, with brain tissue, when you're crossing the blood-brain barrier, you're going to be asked to do some more extensive testing. So think about those things when you, determine your device contact. And use the definitions here. I find them very helpful. And again, keep in mind the discrepancy between the US and the EU. Okay, the other things, so things to think about how this could possibly impact your testing. The thing I see the absolute most is the difference in testing in where the E sits, right? What tests and what endpoints actually have to be evaluated for a medical device. So before, where acute systemic toxicity and material-mediated pyrogenicity was not being requested, then it will be asked for now. That doesn't necessarily mean you have to step into testing. Those are endpoints that can be assessed with some great clinical data, especially if your device is used in a medical setting where the patients are observed for over 24 hours. You have a great leg to stand on to talk about acute systemic toxicity and, of course, a material-mediated pyrogenicity. So again, don't necessarily think you have to run into testing, but step into the conversation about it because it will be expected to be addressed. And again, this documentation, I've noticed a lot, especially with the notified bodies, that if you do not have this initial risk assessment, which at Nelson Labs we call a biological evaluation plan, if you do not have that to lay out your rationale for why you selected what you did, they're going to ask you to go back and do that even after the fact, even after all your testing is complete, because they want to hear your logic, they want to hear your reasoning, and then you want them to get to the same place that you did. So you want to write this like a story in a way, giving plenty of logic and reasoning for all the decision points that you make. And then, of course, asking for the summary report at the end. From my perspective, it does say in the standards that you need an expert to analyze this data. I have had to submit my CV before when I've written a summary report for clients, especially for notified bodies. They want to verify that. They don't give criteria on what an expert is, and I get asked that a lot. I've worked in the testing laboratory industry for over 20 years, and more than 18 of that has been with biocompatibility. But I would say you could, think about higher degrees that are appropriate for biocompatibility, which would be cellular biology, anatomy and physiology, molecular biology, those types of degrees with at least 10 years of industry experience, 5 to 10 years. So think about that for the people that you're setting up to write these reports, set them up for success and make sure that they can be counted as an expert here. Okay, for Part 18, this is the most recently updated standard in 2019. And if anyone has stepped into any extractable leachables testing, you'll know that this is a very unresolved topic of conversation in the industry, especially amongst the laboratories, where there's a lot of different opinions about how to run this chemistry testing. So we're anxious to have a unified standard that brings all of the best practices in one location to provide the best services for our clients. So just a summary here of what's actually changed. So the title change, I brought that up because it's significant. Before, the standard was called Chemical Evaluation of Materials. And it was written almost just like that, with no thought about the application back to the patient, which again is the repeated and overall goal for us here as a laboratory and as manufacturers. So the fact that we're talking about medical devices, so a final finished product, and within a risk management process is also important. Again, allowing us to think about, how does our device contact the patient? Do we really need to do this extensive testing for a surface contact device? If you have a permanent contacting device, I'm going to tell you, you need to live and breathe by this standard. You will need to do extractable leachables testing. It's very important to protect the long-term safety of the patients. Again, we've done a broader definition of chemical characterization, so you can find more language to support what was started in -1. And then a definition that separates extractables and leachables testing. I would say from our chemists, that was the largest complaint. Those are not the same things. Extractables are what is intended or what can come out of the product. Leachables is what is intended to come out during patient exposure. So what can we get to come out of a product? A lot, because we can digest it. But is that really what the patient is going to be exposed to? No. And so we may pull out a ton of things and say, oh, this device is not absolutely safe. But what would the patient be exposed to? Oh, those are actually appropriate levels. So it lets us have that different conversation and assess the device, again, from a risk-based perspective and keeping the patient's safety always in mind. So how do you address this? For all of these different standards that have updated, all the things I've pointed out as discrepancies that could exist between previous standards and regulatory expectations, you do it in a gap analysis is typically how we understand that. So this can come about because you're submitting to a new market. Of course, MDR recertification is a hot topic and on everyone's mind. So questions to ask is always understand your new market requirements, not just what's in the standards, but what's the current climate of the regulatory agency for where you're submitting? What's their expectations? Are they going to accept the chemistry? Do they still expect some lab and animal testing, some in vitro and animal testing to be performed? So again, write a gap analysis. Include your supporting clinical use history. FDA is big on that. They absolutely love to see that data. If it can be communicated in a way that addresses the endpoints, not just, hey, the device is safe, we haven't seen anyone get hurt, but we've not seen any sensitization or irritation claims. We've not seen any fevers, related or noted, with a conversation that ties it back to the endpoints for your reviewer, and that goes a long way. So when we write a gap analysis, we identify, we're going to review our previous testing. We're going to also, at the same time, always review the device that was tested and what the device currently is to make sure we've addressed all of those changes along the way. And then hopefully the dream conclusion is that based on the review and the data, that we can say that the biocompatibility is well supported. And if we can't say that, we'll add in the tests and say with successful completion of the tests, the biocompatibility will be well supported. So I just want to highlight a few things for you to think about and to take away. So track when the standards update, keep track of those. Any 10993 standard is where all the biocompatibility data or requirements are held. Understand the impact on testing, and then provide evidence of risk mitigation, really laying out specifically ways to address and close the gaps for the endpoints that are identified in the standards. And then if you have any additional questions, I think I have a few minutes now that I'll probably take off the side of the stage, but you can also come see me today and tomorrow in booth 2022. Thank you.